Welcome to Stories from the NNI. I'm Lisa Friedersdorf, Director of the National Nanotechnology Coordination Office. Today, it's my pleasure to welcome Pamela Bjorkman, the David Baltimore Professor of Biology and Biological Engineering at Caltech. Pamela, thank you so much for joining us today. To get us started, can you just tell us a little bit about yourself and how you became involved in research and the use of nanotechnology?
Thank you very much for having me. So I guess I got involved in doing research when I was an undergraduate. I majored in chemistry at the University of Oregon, and I discovered that you could not only learn about science in your classes, but you could actually do experiments in labs, and that was just so much fun for me that I've stayed in research ever since then.
So I understand that your research now investigates activity of the immune system. Can you talk a little bit more about the focus of your research?
So your immune system is really very amazing. It can fight off any pathogen that you might be exposed to. And keep in mind that, of course, your immune system can't predict in advance what new virus or new bacterium or whatever is going to come your way. So it has to be prepared to have enough diversity to recognize anything that might come up. Of course, the example is SARS-CoV-2, which is a virus that was not in humans before, 2019. So that's a new virus, and yet our immune system recognizes it and can destroy it. In fact, under the best of circumstances when we fight off that infection successfully. So I'm really interested in how the immune system does this. How does it specifically recognize new pathogens? And so the way we approach this is we use structural biology techniques to actually look at the atomic resolution structures of, let's say, part of a pathogen bound to an antibody, which is one of the protein molecules that your immune system produces. So we look specifically at how antibodies recognize parts of viruses. So until the current pandemic, we'd been working on HIV, on hepatitis C virus, on Zika virus, a little bit on influenza virus, and then we just kind of shifted to concentrate a lot on coronaviruses now. But mainly we're just interested in what are the details of the recognition, and could we use that information to, for example, design a vaccine to prevent an infection or design some kind of therapeutic to actually treat an infection.
So can you discuss the role of nanotechnology in your investigation, this structural biology that you mentioned?
So one of the things we're trying to do is make a vaccine that would prevent infection or prevent disease from a viral infection. And so up until about a year ago, we were actively trying to make a vaccine against HIV, which would progress towards AIDS. And that's really difficult because HIV viruses mutate a lot. And so there are many strains, even in a single infected person. So what we're trying to do for HIV is to improve the immune response using something designed from the HIV virus itself. So I have to back up here a little bit to explain, in general how vaccines work. So basically, what you would do in a vaccine is you have something that mimics a virus, and you inject that into someone, and their immune system thinks it's a virus, but the person doesn't get sick, but the immune system mounts a response against that. And then that response is remembered by your immune system so that if you ever do encounter that virus, your immune system fights it off before it can make you sick. So one of the principles of how your immune system works is that if you have like a multimer of some kind of thing the immune system would see, it mounts a much better immune response. So I'm finally getting to the nanoparticles here. So it turns out that if let's say you inject, some kind of part of the virus, like some individual protein of the virus, and you inject that into an animal model. you'll mount an immune response and the animal will raise antibodies. But if you actually take a nanoparticle and you decorate it with many copies of that exact same protein, the animal will make a much better immune response. And so this is fundamentally very reasonable because your immune system evolved to recognize repeating patterns because pathogens like bacteria and virus have many copies of the same like foreign object on them. So your immune system is really looking for these repeating patterns. So what nanotechnology can do is help researchers make those repeating patterns so that you make like things that look like viruses but are not viruses, so they don't make you sick. So you use these nanoparticles to make repeating arrays of these viral proteins.
So I'd like to ask you a little bit about the makeup of your lab. We often hear that interdisciplinarity is an important aspect of nanotechnology research. And you mentioned in your opening comments that you are a chemistry student and now your focus is in biological engineering. Could you talk about the makeup of your students and researchers in your lab?
Yeah, so I have some students who come to my lab because they want to learn structural biology. So these would be people who are really interested in the chemistry of protein recognition. Like this set of amino acids has atoms that form, hydrogen bonds or electrostatic interactions, or, how do these things recognize each other at the level of atoms? So that's like pure chemistry. So those students end up becoming immunologists and virologists, actually, because there's no way to interpret these structures of antibodies bound to viral proteins in a vacuum. So you have to understand the basic virology of what viruses do and what their life cycles are in order to interpret your structures. And then also you have to understand how the immune system works. And so that's another field of basic biology. So everybody becomes very interdisciplinary because they have to span the basic biology to the biochemistry, which is how to make and purify these proteins for our structural studies. They have to be chemists in order to understand the details of the interactions at the atomic level. And then we have to be nanotechnologists to the extent that we can be to produce these small nanoparticles to then attach whatever we're trying to use to make the immunogen. And so it's very interdisciplinary. We also have an effort in liquid handling robots for automating assays in the lab. So we have a lot of things that are fairly tedious to do, binding assays and so on. And so we have people in the lab who are experts in programming liquid handling robots so that they will basically pipette amounts of liquid up into different plates and multi-well plates and so on to do these assays in an automated way. So these days, to do this type of research, students and postdoctoral fellows have to span a lot of technologies. And that's somewhat easier to do at Caltech because we're a small place. And so we have a lot of cross-disciplinary research with engineers and biological engineers, more conventional engineers, and also biochemists and biologists.
I understand you've been at Caltech for a while, and I believe you were there when Clinton first announced the concept of a national nanotechnology initiative over 20 years ago now. And I wonder if you have thoughts on where you've seen nanotechnology progress and where you think the most exciting applications are into the future.
That's a great question. I actually have gotten help on this through Julia Greer, who's a professor at Caltech in engineering and applied sciences. In talking to her and in hearing her talk, she's making a lot of structures at the nanoscale. And so from talking to her, she would say that the most important advances would be miniaturizing things, miniaturize the components of your phones and cameras and computers and detectors and so on, And then you can also, through this type of nanotechnology, have amazing computational power. And these things, of course, are interrelated because these computational capabilities that we have now are just amazing. But what they're allowing us to do is to predict properties of materials and their behaviors. And then that in turn allows more research and understanding of how to miniaturize yet other things that will go back into the computational things. And so there's a great future ahead in this. You can imagine all kinds of things being done. Now, my research is not in that type of nanotechnology because what we're basically doing is we're using nanoparticles that are made from protein components. And so there's a whole nother engineering aspect, the type of things that Julia Greer does with non-protein materials. But I think there's an amazing future, and things that have already been done have really revolutionized our way of life.
You talked about the interdisciplinarity in your group and also when students come to you for interest in specific areas, they can be part of your team focused on everything from liquid automated robots or looking at the details of structural biology focus of proteins. When you interact with, I would say, especially undergraduates, what advice do you give them if they are interested in working in this field?
The advice I would give them is you need as basic a set of knowledge as you can get. So a lot of things that you learn as an undergraduate may not seem applicable if you decide, okay, I want to do nanotechnology, I want to be an engineer, and I want to make new materials and so on. But you really need a very strong background in chemistry, you need it in thermodynamics, you need it in math and physics. And that's exactly, of course, the sort of background undergraduates get at Caltech. But you have to hang in there to learn all these basic things so that later you can specialize in something that might be more what you really want to do as a goal. But you have to kind of suffer through all this stuff so that you can develop a tool set so you can tackle really multifaceted, complex problems that require interdisciplinary knowledge.
Pamela, thank you so much for taking the time to talk with us today. Do you have any closing thoughts that you'd like to share with our listeners?
Well, I think it's a great time to be doing science. There are so many ways to contribute to making our world a better place. So I would really encourage people to follow their dreams if they want to be a scientist. There's so many opportunities. I just think it would be great. The more people that get involved in science at a young age, I think it will help us with the world's problems, which right now I see as the current pandemic, obviously, and then future epidemics and pandemics, which are likely to happen. And then also climate change. And these are related to each other because as humans change the climate, we are sort of changing the habitats of animals, and those animals are transmitting viruses to us. So everything is kind of interrelated, and the more people can learn to do the science that would help with these problems, I think the better our world will be.